for ecosystems that lean on deer, and for the uneasy border between species. Zombie deer, chronic wasting disease, CWD. Walk into a frosty November morning in Wisconsin and you'll see the stakes emblaze orange. Families set up camp, brew coffee on tailgates, and tell the same jokes their grandparents told. Then someone mentions the map, colored dots spreading across counties, and the laughter thins. Chronic wasting disease is here. It doesn't make deer snarl or crave brains. It makes them fade, ribs showing, heads drooping, saliva threading from the mouth. A hunter tracks a buck for hours, takes a clean shot, and then wonders if he just harvested a question mark. The action isn't a single outbreak, it's a slow creep. First identified in captive mule deer in Colorado in the late 196s, CWD moved into wild herds and then across state lines like spilled ink. Today it's confirmed in multiple states and Canadian provinces, plus parts of Scandinavia. The transmission web is messy and efficient. Infected deer shed prions in saliva, urine, and feces long before they look sick. They nuzzle, lick, share scrapes, crowd salt licks, and everything they touch becomes a potential handoff. Carcasses magnify it. Scavengers drag parts. Soils inherit seeds. Science chased the spread with better eyes. RT-QuIC and PMCA, lab assays that amplify seeding, made it possible to detect tiny amounts of prion activity in tissues and fluids. Lymph nodes from the head and neck, tonsils, and even feces can carry enough seed to light up a tube. Field teams collect samples from hunter-killed deer. Labs shake tubes and read the curve within hours to days. A strong signal doesn't just say infected, it says infectious potential, and that changes what families do with a freezer full of meat. Meanwhile, researchers press the species barrier from the lab side. Rodents engineered with cervid PRP catch CWD readily. Mice with human PRP resist far more. Studies in macaques split nerves. Some showed no disease after years of exposure. Others found subtle pathology or biochemical signals after oral or intracerebral dosing with CWD material. The takeaway isn't doom, it's uncertainty with teeth. There's a barrier, but it isn't a wall you bet dinner on. Agencies repeat the line that matters. Don't eat meat from a positive animal. Period. Curiosity gap. If a barrier slows spread, what happens when dose and route change? Policy tried to slow the dance. Feeding and baiting bans aimed to stop deer from clustering unnaturally. Carcass transport rules kept spinal cords and heads, the prion-rich parts, out of gas station dumpsters across borders. Check stations popped up like seasonal pop-up stores. Hunters learned a new ritual. Bag the retropharyngeal nodes, drop them at the tent, wait for a call. If the test is negative, you wrap and label. If it's positive, you don't take a chance. You don't feed it to pets. You dispose of it per rules that read more like biohazard than barbecue. The human beat is small and private. A father and daughter team drags a 10-point buck to the truck, high fives, then gets quiet when they fill out the sample card. They drive home, hang the deer in a cold garage, and check their phones every hour for 5 days. When the result pings negative, relief is loud. When it pings positive, the garage becomes a triage site. Gloves come out, knives get segregated, meat goes into bags they never thought they'd use for food. The daughter asks if they did something wrong. The father says no, they did everything right by checking. Meanwhile, the woods don't care about feelings. Prions ride out winters. Certain clays bind PSC and can even boost its uptake when deer root and graze. Plants may wick up particles on surfaces, making the interface between soil, leaf, and tongue a quiet conveyor. Land managers argue over culling versus targeted removals, over whether reducing density slows spread or just breaks traditions. Ranchers with captive herds face business-ending decisions when tests find a hot pen. Scientists push for nuance without murk. There's no evidence CWD transmits to humans through casual contact. Hunters who test and follow guidance dramatically cut their risk. Cooking doesn't fix contamination. Knives and grinders need segregating and cleaning with prion-aware protocols. Heads, spines, and offal shouldn't ride in coolers to cabins 2 states away. These aren't abstract rules. They're speed bumps that actually slow an epidemic measured in seasons, not days. The humor you cling to at camp gets drier. Somebody taps the bleach bottle and says it needs a meaner cousin. Somebody else jokes about gossiping molecules at the salt lick. The jokes aren't denial, they're armor for the waiting. What nobody knew when CWD first appeared in pens is how long the landscape can hold a memory. Now we do. It changes how we hunt, butcher, share meat, and talk to kids about why their deer needs a test before it becomes chilly. It also hands hospitals a mirror. If soils can keep a code, so can steel. The next chapter walks into operating rooms and asks a blunt question. When medicine forgets that shape can travel, who pays? When medicine forgets, iatrogenic transmission. Unknown to them, across town, a different kind of hunt was underway. Inside a hospital, on polished steel. A neurosurgeon finished a delicate biopsy, rinsed an instrument set, and sent it down to sterile processing. The trays came back gleaming. Days later, someone said the word nobody had put on the chart yet. CJD. A shape might have just taken a ride to another patient. Hook. How do you stop a memory that clings to metal? The stakes are trust and time. People walk into hospitals believing the tools are clean in the way that matters. With prions, clean needs an asterisk. In the 197s and 80s, children needing growth hormone got pituitary extracts pulled from cadavers. A few vials carried a hidden code. Years later, some of those kids, now adults, developed creutzfeldt-jakob disease. The dose had been tiny, the route was direct. The timeline felt cruel. Dura-mater grafts did it too. Patches taken from deceased donors, treated but not prion-killed, were used to repair skull-based defects or cover the brain's surface. Some patients, years after routine surgeries, began to stumble, jerk, and forget. A handful of corneal transplants passed the shape through an eye, and rare clusters traced to neurosurgical instrument sets that looked spotless but held a template in their scratches. Each case forced hospitals to rewrite muscle memory. Here's the action, not the myth. Standard sterilization wrecks bacteria and most viruses. Autoclaves run at 121 degrees Celsius for 15 minutes, steam under pressure, and the bugs die. Prions aren't bugs die. Prions aren't bugs. Amyloid folds pack so tight that water and enzymes can't get teeth in. Dried onto steel, the seeds adhere like barnacles. A quick brush, routine detergent, and a normal autoclave cycle leave enough structure to matter. That's how a template moves from one skull to another. So the decon reality changed. When brain or spinal cord might be involved, biopsies, deep ENT cases near lymphoid tissue, pituitary and posterior eye work, you don't trust the usual. Protocols call for concentrated sodium hydroxide or high-strength sodium hypochlorite soaks, followed by extended autoclaving at higher temperatures and longer times. Some instruments don't survive that. That's a feature, not a bug. For certain high-risk steps, single-use tools are now the norm. Path labs swap blades and change traps before opening a suspected CJD case. Endoscopes that touched lymphoid-rich tissue get flagged and sequestered pending diagnosis. Meanwhile, what nobody knew at the time of the first iatrogenic reports is how much behavior needed to change on paper. Disclosure policies matured. If an instrument set might have been exposed before a diagnosis, infection prevention teams track where it went and who it touched. Patients get calls nobody wants to make or take. The counseling is blunt and careful. The absolute risk is low but not zero. There's no screening blood test, there's no post-exposure pill to swallow. The goal isn't panic, it's respect for uncertainty. Blood services made their own moves. People who lived for long stretches in places and times with high BSE exposure were deferred from donating. It wasn't a verdict on them, it was a gatekeeping choice to keep a shape off the highway. Tissue banks mapped donors more tightly. Dura and cornea sourcing shifted to systems that minimize unvetted material. Growth hormones switched from pooled human extracts to recombinant products. No more cadaver in a vial. Here's the humor beat that keeps you from grinding your teeth. The stain that laughs at bleach, and then invites friends, shuts up when bleach shows up with sodium hydroxide, extra heat, and a calendar. It's chemistry, not vibes. Inside operating rooms, micro-stories stack up. A scrub tech holds an instrument and asks the circulating nurse to check the case notes again. A pathologist pauses before cutting and swaps cassettes. A resident learning their first brain biopsy also learns a new alphabet of warnings taped to the wall. NaOH here. Do not reuse there. Quarantine tray 3 if prion suspected. None of this makes surgery scarier. It makes it smarter. Impact ripples out. National guidelines now spell out risk tiers by tissue. Brain, spinal cord, posterior eye, and pituitary at the top. Lymphoid-rich mucosa and dorsal root ganglia in the middle. Blood and most peripheral tissues far lower. Hospitals segment instrument inventories so a possible exposure doesn't contaminate the whole fleet. Documentation got less romantic and more exacting. Lot numbers, tray IDs, patient labels that follow a path like airline luggage with better ethics. And still, there's residual risk. Sporadic CJD can look like many things for weeks. A patient may enter the system labeled rapid dementia or rule out autoimmune encephalitis. Only later does an MRI pattern or a cerebrospinal fluid RT-QuIC result light up the prion path. That lag is where iatrogenic stories begin. The fix isn't clairvoyance, it's default caution for certain tissues and procedures, plus fast pivots when suspicion rises. The counseling lands in a human place. A surgeon sits with a family and explains that an instrument possibly touched high-risk tissue before the diagnosis was known. The chance of harm is very small. The hospital will monitor. There's nothing to do today but live your life. The family nods, asks hard questions, and then goes home to a dinner that tastes different. Trust bends, but doesn't have to break when systems tell the truth. What this chapter proves is simple and uncomfortable. Shape travels on steel if we let it. The countermeasure is choreography, chemistry, heat, segregation, and paperwork that reads like a shield. And remember the woods? Those same principles. Don't move heads and spines. Don't mix toolkits.
Spontaneous
Don't
errors.
guess. Apply
Sporadic
to fluorescent
CJD
light.
and aging proteomes.
3 days after the hospital scare, a different story walked in without any exposure at all. No transplant, no surgery, no deer camp. A retired school principal started telling the same story twice at dinner. A month later, she forgot the route to a grocery store she'd driven for 20 years. Her daughter said stress. Her son said grief. The ER said possible stroke. The MRI said something else. Diffusion changes in the cortical ribbon, bright where brain should be quiet. The shape had arrived without an invitation. This is the villain nobody can point to. Sporadic creutzfeldt-jakob disease shows up in about 1 person per million each year worldwide. There's no known trigger you can circle in red ink. It's a roll of molecular dice in a body where, with age, the table tilts. Proteins spend their lives folding, refolding, and getting cleared. That balancing act, proteostasis, runs on chaperones, proteasomes, lysosomes, and autophagy. With age, the crew tires. Oxidative stress scuffs surfaces. Heat shock responses lag. The same quality control system that once turned misfolds into confetti now lets a few slide by. Somewhere in that latency, a single PRP molecule may flip into a rare conformation. On most days, it snaps back or gets eaten by the cleanup squad. On a bad day, it fits against a neighbor just right and teaches the wrong pose. From there, it's the same kinetics you've seen. Growth, fragmentation, more teaching. No deer, no donor, no instrument, just time and chance pushing a molecule across an energy ridge. Clinically, the slope is steep. Families talk about a good week that already feels off, then a month where speech shortens, attention frays, and myoclonus, those sudden jerks, startle everyone at the table. Gait goes shaky, vision blurs, sleep grows strange. A neurologist asks for tests that sound like alphabet soup. CEG, MRI, CSF, RT-QuIC. The EEG sometimes shows periodic sharp wave complexes, spiky patterns that whisper prion. The MRI's diffusion-weighted images can flash bright in the cortex or deep nuclei, mapping the spread like heat marks on a wall. Cerebrospinal fluid, dripped through an assay that reads seeding RT-QuIC, lights up with growing fluorescence as PRP in the tube learns the wrong fold. It's not a horoscope, it's kinetics on camera. Meanwhile, the differential diagnosis is a crowd. Autoimmune encephalitis can sprint, viral encephalitis can confuse, toxic metabolic crashes can mimic. Doctors throw antibodies and antivirals when those lanes seem likely, because they're the lanes with exits. But when the pattern holds, rapid dementia, myoclonus, a kinetic mutism creeping in, CJD comes to the front. A brain biopsy could confirm, but often, with imaging and RT-QuIC, it's not needed. Families already have the answer they didn't want. Stakes land with paperwork and kitchens. There's no cure. Median survival is measured in months. Caregivers quit jobs. Siblings fly in. Someone Googles at 2 a.m. and stares at the words spongiform change and pictures holes. The better picture is synapses dimming, networks failing, a city losing power block by block. Hospice becomes a plan you make while still setting coffee for the morning. Advanced directives jump from theoretical to urgent. Durable power of attorney, do not resuscitate orders, who knows passwords, life admin as love language. Here's a micro story that makes the science feel human. The principal had a favorite sweater she wore on exam days. Blue, soft at the elbows. In week 6, she reached for it and asked whose it was. In week 8, her son put it on her shoulders and she smiled anyway, without recognition, because kindness still made sense to a nervous system that couldn't name it. The disease takes nouns first, then verbs, then the quiet glue between people. Families learn to meet presence without words. What nobody knew in the 199s, but we lean on now, is how much earlier we can read seeding. RT-QuIC turns suspicion into evidence without a scalpel. A few drops of CSF, hours of shaking in a plate, and the curve climbs if seeds are there. Not perfect, but precise enough to change how we talk. It also sharpened infection control when cases present as unknown encephalopathy. Labs flag, instruments quarantine, and the iatrogenic stories you just heard get rarer. Genetics still hangs in the background. PRNP codon 129, methionine or valine, doesn't just dictate familial outcomes, it shapes sporadic risk and phenotype too. Methionine homozygotes are overrepresented in certain SZJ subtypes, and incubation in variant disease tracks that background. It's not destiny, it's a tilt. Aging proteomes slide faster on some slopes than others. Treatment trials come and go with headlines that fade. Anti-aggregation compounds, conformation-specific antibodies, copper-binding ideas, chaperone boosters. None have moved the clinic needle yet. The timelines are crueler than our patience. Still, early detection tools matter. If a therapy that slows seeding ever clears the bar, catching the disease when seeds are few will buy time you can feel. Impact ripples into law and money. Families scramble to secure disability benefits, protect savings, and guard against fraud targeting vulnerable elders. Social workers become navigators. Palliative teams manage symptoms. Myoclonus with clonazepam. Sleep with careful routines. Anxiety with presence more than pills. Swallowing assessments prevent aspiration. Beds get rails, rugs go away. The home becomes a controlled landscape, not a museum of the past. Meaning is the last hard chapter here. Sporadic CJD reminds us that randomness is a player in our lives, not a footnote. You can do everything right and still meet a bad fold. The response isn't superstition, it's intention. Advance directives while you're well. Conversations that scare you a little now, so they hurt less later. Donations to brain banks that turn endings into data futures can use. Dignity isn't a cure, but it's a technology. It makes the final months less brutal for everyone in the room. And then there's presence. Families mark last good days. They plan small rituals, a favorite song at lunch, a brief walk at sunset, a call with an old friend on speaker so the voice can run across the room. The disease writes its timeline. People write around it, leaving notes for themselves in the margins so they remember the person before the template took their words. Beyond prions. Prion-like spread in Alzheimer's and Parkinsons. While families faced the speed of CJD, another set of families were living a slower heartbreak. A grandmother forgot recipes she'd made for 40 years. A former engineer's hands began to tremble, then curl, then refuse the buttons on his shirt. Doctors said Alzheimer's, Parkinsons, familiar names with unfamiliar mechanics. Here's the careful line. These aren't infections like Kuru or variant CJD, but inside the brain, something eerily similar happens. A misfolded shape teaches neighbors the wrong pose. Start with Alzheimer's. 2 proteins drive the discussion: beta amyloid and tau. Beta amyloid, a chopped fragment from a larger precursor, aggregates into plaques outside neurons. Tau, a microtubule stabilizing protein, misfolds inside neurons into tangles. The mystery wasn't that they accumulated, it was how the pattern spread. Brock and Brock mapped tangles marching from the entorhinal cortex through limbic circuits and into neocortex, stage by stage, as if following wiring diagrams. That map looked less like random rot and more like a route. In lab dishes and animal models, seeds of misfolded tau or amyloid injected into one region trigger pathology that appears later in connected regions. It's not replication in the infectious sense, it's templating, seeding mechanics echoing prions without the human-to-human epidemiology. The seed lowers the energy barrier for the next copy to match its shape. Neurons ship cargo along axons, vesicles, bud, fuse, and leak. Exosomes and synapses act like ports. The misfolded core catches rides the cell built for work, not sabotage. Now slide to Parkinson's. The key protein, alpha-synuclein, normally helps with synaptic vesicles. Misfolded, it assembles into Lewy bodies. Clinicians noticed a strange prodrome: constipation, loss of smell, REM behavior disorder, acting out dreams, years before tremor. Brack staging again traced a path, this time from the olfactory bulb and gut up the vagus into midbrain nuclei where dopamine neurons live. In animals, alpha-synuclein seeds placed in gut walls later show up in brainstem, cut certain nerves, and the spread slows. The phone tree metaphor holds. Here's what nobody knew in the early Alzheimer's era. You can read this templating in tubes, like with prions. RT-QuIC variants now detect alpha-synuclein seeding activity in cerebrospinal fluid, and in research settings, even in skin or olfactory mucosa. For tau, seeding assays exist too, capturing the lag-then-surge curve that betrays templating. The word prion-like lives here carefully to describe the mechanism, seed and template, not the epidemiology. There's no credible evidence that casual contact spreads Alzheimer's or Parkinson's between people. The spread is within one brain circuits. Boundaries matter. Classic prion diseases cause transmissible spongiform encephalopathies with well-documented iatrogenic and dietary routes in rare contexts. Alzheimer's and Parkinson's are not like that in everyday life. Still, scientists pay attention to edge cases. Old duramater grafts, the very kind linked to iatrogenic CJD, have produced patients with beta amyloid deposits at surgical sites years later. That doesn't mean their dementia traveled in. It means amyloid can seed in tissue when placed there. It's a caution about instruments and tissues, not a cue to fear handshakes. The stakes of the prion-like frame are huge. If misfolded tau or alpha-synuclein spreads by templating along circuits, interventions can target the handoff. Antibodies that bind the misfolded species might soak up extracellular seeds. Small molecules could cap fibril ends, so growth stalls. Peptidomimetics might wedge into the beta sheet zipper and jam it. Chaperone inducers could boost the cells, refold and destroy Kuru before the avalanche starts. And because staging maps predict which regions go next, clinicians can time treatments to circuit vulnerability, not just symptoms.